You get DNA, and you get DNA, and you get DNA. And this is Norris Teach Science, starring Mr. Norris. We're doing another biology edition of the video. Today we're going to talk about DNA structure and DNA replication. So that by end of this video, you can say, I can explain the structure of DNA and how it works to replicate itself. So let's jump into what is DNA. I've mentioned this in many videos about DNA, DNA's in the nucleus, all this other stuff. So DNA stands for deoxyribonucleic acid, DNA. That's where it comes from. It's an abbreviation. DNA is also known as a nucleic acid. That means it has the same components as what the nucleic acid when we talked about in the macromolecule organic molecules videos. That means that its monomer is what? Did I hear you say nucleotide? Because that's what it is. The sugar in DNA is deoxyribose because nucleotides are made of a phosphate, a base, and a sugar, and the sugar for this one is deoxyribose. The job of DNA is to hold genetic information and hold the instructions to making proteins. Very important that it has the instructions making proteins because proteins lead to all the traits that you have. So proteins are like the reason why you say have green eyes, if you have green eyes. I personally have hazel eyes. So as you can see in this picture, this kind of is what the DNA would look like if we sketched it out. So let's talk a little bit more about the structure. The structure is called a double helix. It's like a ladder that you twist. You can kind of see that in the picture on the side. It's made of nucleotides, like I mentioned. Each nucleotide has a base, sugar, and a phosphate. The bases are on the inside of the DNA strand. They're on the inside, right here. The hydrogen there's hydrogen bonds connecting the two bases on each side. The phosphates and the sugars are on the outside of the base, and they make up what we call the backbone of it. So if you think of it like a ladder, here's a ladder. These outside vertical lines would be the phosphate and sugars, and the inside horizontal lines are the bases. Now, speaking of the bases, there are 4 different nitrogen bases: adenine, thymine, guanine, and cytosine. A, T, G, C. So you can, when you do this, you can just say the letters. Most times when people are talking about DNA, if you just say A, they know what A means. But they do pair up a certain way. So A's will pair with T's and T's will pair with A. Someone once told me that if you think apple in the tree, A and T, that's how you can remember that. And G will pair with C and C will pair with G. Someone once told me car in the garage. Car for C, garage for G, they go together as well. A will not pair with G or C, and vice versa. They only pair in this certain way. Now, what is DNA replication? DNA replication means to make an exact copy of itself. We talked in the cell replication videos about DNA copying itself, and it's really important. So this is the process that happens. Now, why do we need DNA replication? Just referring back to cell division, that especially in mitosis, cells need to have 2 complete sets of DNA to give its daughter cells when a cell divides, for mitosis, meiosis, all that stuff. And when does it happen? Referring back to our cell division stuff again, it happens during S phase of either mitosis or meiosis. In interphase, remember both of those have interphase. It happens during S phase of interphase during mitosis and meiosis. Well, how does this work? So DNA replication, step 1. What happens is, remember, it's a twisted ladder, a double helix. What first happens is that it untwists. And this enzyme called helicase comes in, and it's like a pair of scissors, and it snips the hydrogen bonds between the bases. So the DNA starts to split into two sides. After that, new nucleotides move in and they match with their appropriate bases. So if there's a G here, a C comes and matches up. C with G, T, A, T, stuff like that. They come and match up. They're not attached yet. They have just matched up. Now, the third step is this enzyme called DNA polymerase comes through, and it attaches the bases together with hydrogen bonds. There's a little bit more to this about the direction it happens, about 3', 5', and 5', 3'. But for this instance, we're just going to cover the basics of it. But right now, you just need to know that after the bases match up, they are bonded with hydrogen bonds with DNA polymerase. And the end result, there's two complete molecules of DNA that now exist. So you have two sets of DNA. One side of each was from the original DNA strand. You can see that here. Stars and pound sign. Each of the new ones have one of those, and then the other side is the newly formed side of the DNA. And this is all happening all at once. Let's practice this. One thing you're going to have to do is you're going to have to be able to look at a DNA strand and give the other side. So if we look right here, we already have split it, cut the things up, and we're just going to match up the bases. So what you would be told was write the complementary side of the DNA strand. So here's the original DNA strand here. We're going to write the other side. If it says A, we're going to write T. T, we're going to write A. C, G. T now goes to A, and we just continue on as we go along. A, G, G, T, A, C, C, T, G, A, T, C. All right, I did one side. What I want you to do, pause the video real quick. You have the other strand of DNA that you need to write for as well. Pause real quick. I'll give you some time. 5, 4, 3, 2, 1. All right. So I am not going to write it because all of this that you see up here would be written right here, because remember, they're making exact copies of DNA. So I hope you understand this. Like I always say, there's more out there about DNA. We're going to cover a couple more things with it in this video section. But if you need to know more about DNA, you want to know about 3', 5', 3' replication, all that stuff, just go to Google and you can search it all up. But like I always say, hope you learned something. And this is Mr. Norris, and he is signing out.